As part of NOAA's Oyster Reef Ecosystem Services project, we conduct field sampling in the Chesapeake Bay. Field sampling involves trapping fish on different habitat types. After 24 hours, we retrieve the traps and count and measure the different species of fish. We place half the traps on restored oyster reefs and half on unrestored oyster reefs, and then we compare the data from the two sites.
White perch, black sea bass pot, 300.
Our work at CERC is looking to understand the fish communities that are using restored oyster reefs. And this imaging sonar, or multi-beam sonar that we're using, allows us to understand what fish are doing when there's no trap in the water and how abundant they are and what their behaviors are.
Recreationally and commercially important species are red drums and your striped bass, things that we like to eat. Are they gaining significant nutrition from the oyster reef, and can we quantify that? And then follow them as the reefs mature. So how do those fish communities change over time?
We are looking at fish and crab use of restored oyster reefs in the lower Chesapeake Bay. So we can take this piece of equipment and embed it into the oyster reef and look at what organisms settle on the oyster clumps that are actually going to be inside of here, flush with the surface of the oyster reef.
The LBV, little benthic vehicle, actually has a color camera, black and white camera. It's got infrared lighting, a navigation system. It has a laser for scaling, for getting an estimate of size, and it's got side scan sonar that we can position at the reefs and actually quantify the number of crabs feeding on the reefs, and then we link that with abundance information to truly get an assessment of how much more production of blue crab we're getting by the reefs.
We'll be sampling 5 times a year, each year for the next 3 years. We'll have several things that we learn along the way, and then at the end we'll be able to come back and look at that entire data set and assess the benefits that have been provided to date by the restoration effort.
We're really trying to get a snapshot of the whole ecosystem to see how these restored oyster reefs affect a larger scale community.
We believe a mature oyster reef is going to have a lot more water quality value than when they're first planted and when they first start. Divers take the bottom tray off of this and we leave it out there for a month. We bring this back in the laboratory, attach a stirring top like this, and we're able to simulate nature in the laboratory under controlled conditions.
Ecosystem modeling is useful for helping us to understand the bigger picture. You can't measure everything everywhere and project out into the future, so you use models to estimate what will happen in the bigger picture.
Oysters are these natural engineers in the estuarine environment. They have a really key place in structuring the community of organisms, as well as affecting the chemistry in the water column.
We're creating reefs that are doing some really special things, and we think that they are the engines of the estuary.
Working together with the other researchers within the NOAA-ORES project allows us to each get different kinds of data that come together to form a much better picture of what's going on within the oyster restoration projects.
We're hoping that our results can be used by decision makers to figure out what types of reefs perform the best so that we can restore reefs as efficiently as possible in the future.
The research that is done by NOAA and its partners, both in the lab and in the field, is important because it allows us to put a value on oyster restoration in the Chesapeake Bay.